Hi guys, welcome to my YouTube channel. Don't forget to subscribe and share.
Hi, Assalamualaikum. Alright, for today's class, we're going to continue with subtopic factors affecting enzymatic reaction. So in our previous class, okay, we know that effect has 4 factors that will affect enzymatic reaction. Okay, first is the substrate concentration. Okay, when the substrate concentration increase, the rate of reaction increase until all the active site of enzyme is fully saturated with the substrate. So the rate of concentration, the rate of reaction will not increase. Okay, second one, okay, we have the temperature. So as you know from the graph, when the temperature increase, the rate of reaction increase due to the kinetic energy of the enzyme and also the substrate increase. So it will increase the chance of collision where the substrate going to bind with the active site of enzyme. Okay, so the rate of reaction will increase until reach optimum temperature. Above the optimum temperature, the rate of reaction will sharply decrease because the enzyme start to denature. Okay, the enzyme denature due to the break off of the excessive bond that maintain the globular shape of the enzyme. The bond is hydrogen bond, ionic bond, disulfide bridge, hydrophobic interaction, and also van der Waals forces. Okay, and for the third factor affecting enzymatic reaction is the pH. So as you can recall, okay, every enzyme has their particular range of pH. Okay, every enzyme is their own particular range of pH. Okay, for example here, pepsin, the pH, the particular pH that the pepsin act very actively or efficiently is in acidic condition, which the pH is 2.0. For amylase, the preference pH is almost natural pH, which the pH is 6.8. While the arginase, the preference pH is basic, which the specific reading is 7.6. Okay, so in different pH range, what happened to the enzyme? Enzyme will also denature because the reaction between the R site, okay, between the R group, okay, it will cause the excessive bond, okay, the hydrogen bond and also the ionic bond to be break. So the enzyme will cannot maintain its shape, so that it cannot conduct catabolic reaction and also anabolic reaction. Okay, so the next factor is cofactors. Okay, so cofactors. What is cofactors? Okay, cofactors can be formed of metal ion or small organic molecule. Okay, it is in form of metal ion or small organic molecule. And this cofactor, it required in the small amount. For what? To make sure the enzyme can act more active. Okay, can act more efficiently. Okay, so this cofactor, okay, the enzyme are known as simple enzyme when they functional form consists of protein with tertiary structure. So with tertiary structure. So this tertiary structure is referred to the cofactor. And this cofactor is required in a small amount, okay, so that the reaction of enzyme is more effective or efficient. So this cofactor can be formed of metal ion or small organic molecule. Okay, so for cofactor is a non-protein component required for some enzyme to function. So remember, enzyme is protein, but cofactor is non-protein. So this is considered as organic, okay, can be organic or inorganic. So this cofactor, it can be divided into 3 types: metal ion activator, coenzyme, and also prosthetic group. Okay, metal ion activators, coenzyme, and also prosthetic group. Okay, so here is two component of enzyme. So as you know, enzyme has active site and also allosteric site. Active site is the site which will contact with the substrate, where the enzymatic reaction occur. Okay, so other site is called as allosteric site. Okay, if the enzyme without cofactor, we call it as apoenzyme. Okay, if the enzyme without cofactor, we call it as apoenzyme. Enzyme with cofactor is called haloenzyme. Holoenzyme. Apoenzyme plus cofactor is holoenzyme. Apoenzyme, enzyme without cofactor. Holoenzyme, enzyme with cofactor. So this cofactor, it will bind to the allosteric site lah, means that the part of the active site. Okay, so here again, apoenzyme plus coenzyme, okay, become haloenzyme. So this is example of cofactor. Okay, prosthetic group is cofactor. Metal ion is also cofactor. Coenzyme is also cofactor. Okay, so for the first type, metal ion activator. Okay, so obviously they are metal ion. So this metal ion, how it bind to the enzyme? It will loosely and temporarily bound to the enzyme by weak ionic bond. Okay, remember Okay, all these cofactor, they may bind to the enzyme. But you need to remember the types of binding. Okay, and then the binding is permanently or temporarily. Okay, in this case, for metal ion, it will bind to the enzyme loosely and temporarily. How it binds is by forming a weak ionic bond. So this weak ionic bond can make the metal ion bind to the enzyme and easily to break with the enzyme. So this metal ion help draw electrons away from the substrate molecule, making bond less stable and easier to break. So example of metal ion, okay, example of cofactor metal ion, okay, please remember magnesium ion and also calcium ion. Okay, here is the two common metal ion which act as a cofactor. Other than that, ni lah, okay. Alright, example. So salivary amylase. Okay, the amylase enzyme, it's required a chloride ion. Okay, it's required a chloride ion to convert starch into maltose efficiently. And for the thrombokinase, the enzyme which play a very important role for blood clotting, is required calcium ion to convert prothrombin into thrombin. Okay, here is the example requirement of cofactor, the types of metal ion to help the enzyme act more effectively. Okay, and here also the example. Okay, and again, this one show here is the enzyme. Since there is no coenzyme, so this is called as apoenzyme. And then the cofactor here, metal ion, bind to the enzyme. So the enzyme now called as holoenzyme. So now the binding of metal ion, it trigger the enzyme to act more effectively. Right. So that the substrate can bind to the enzyme and then the enzymatic reaction occur to produce the product. So here, an enzyme is unable to become functionally formed unless metal ion is available. Once the metal ion is bound, the active site form and enzyme is ready to carry out its function. So it shows that how important the cofactor to the enzyme. Okay, second one, okay, is coenzyme. Okay, coenzyme is organic substance and it will bound to the enzyme loosely and also temporarily, okay, by forming hydrogen bond. Compared to metal ion, yes, it bound loosely and temporarily, but by forming weak ionic bond. But this coenzyme, they bound to the enzyme loosely and also temporarily, but they may bind to the enzyme by forming hydrogen bond. So it will readily detach from the enzyme. Maksudnya lagi mudah untuk dia keluar daripada enzim tu. So the function is to transfer electron or part of substrate from one molecule to another. Transfer electron or part of substrate from one molecule to another. So most water-soluble vitamin are coenzyme or component of coenzyme. Fat-soluble vitamins such as vitamin A, D, E, K are not involved as coenzyme. So water-soluble vitamin only. Fat-soluble vitamin, these are they, is not involved as coenzyme. Okay, so here is the example of coenzyme which act as the cofactor, okay, the coenzyme NAD plus. Okay, NAD plus is a short form of nicotinamide adenine dinucleotide. Okay, so this is derived from nicotinic acid, a member of vitamin B complex. Act as a coenzyme to hydrogenase by acting as hydrogen acceptor. So the enzyme is dehydrogenase. Okay, so the dehydrogenase enzyme required coenzyme NAD plus to make sure it can function more effectively. Okay, so here is other example NAD plus, ATP also coenzyme, okay, which help in transfer of phosphate group. Coenzyme A, okay, help in transfer groups derived from organic acid. Okay, here is other example for coenzyme. Alright, so the last types of cofactor is prosthetic group. Okay, for prosthetic group, they are organic molecule and they will bind to the enzyme by forming tightly bound and permanently attached to the enzyme. So ingat they bind tightly and permanently to the enzyme, okay, by covalent bond. So covalent bond is a very strong bond, so that's why it can bind permanently. Example of prosthetic group is the heme, okay, in cytochrome, and this heme is bind in cytochrome oxidase. Okay, the function is to transport electron to produce ATP in cellular respiration. Okay, so for prosthetic group, the example is heme. Okay, next slide. Okay, here is the example lah. Okay, heme as prosthetic group. So, okay, here is the structure of here is the structure of the heme. Okay, and then here is the function of cofactor. Work by binding with the enzyme, sometimes alter the shape of enzyme so that the enzyme can bind more effectively with the substrate. Okay, and then enzyme activators, okay, inorganic ion or metal ion. The function is to draw electron away from the substrate molecule, making bond less stable and easier to break. Okay, and for coenzyme type, the function is to transfer electron or part of substrate from one molecule to another. And for the prosthetic group, the function is to carry of atom or electron. So why transferring one compound to another in metabolic pathway? Because none of amino acid side chain is good as electron acceptor. Okay, so here the green one is referred to the function of particular types of cofactor. Okay, we have metal ion, coenzyme, and prosthetic group. Okay, that's all for cofactor. Next, we're going to continue with subtopic 4.3 inhibitor.